What is mitosis? Mitosis is the process of cell division. It produces identical copies of cells and is involved in growth, cell repair or cell replacement, and asexual reproduction. When cells divide by mitosis, the number of cells increases. This causes the organism to grow as a result. In humans, each cell normally contains 23 pairs of chromosomes for a total of 46 chromatids. In this video, we will look at what happens to 4 pairs of chromosomes. The cell cycle has two major phases, interphase and mitosis. Interphase is the phase of the cell cycle in which a typical cell spends most of its life. Interphase is divided into 3 phases, G1, S, and G2. During all 3 parts of interphase, the cell grows. The nucleus and nuclear envelope are distinct, and the chromosomes are in the form of loosely arranged, thread-like chromatin. In interphase, the nucleus replicates its DNA and centrosomes. The chromatin starts to coil in the nuclear membrane as the cell transitions from interphase to prophase. Next, we begin mitosis. Mitosis is the part of the cell cycle in which replicated chromosomes are separated into 2 nuclei. Mitosis involves 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. The first phase of mitosis is prophase. Prophase is where the chromosomes in the nucleus really condense and coil. Each chromosome consists of 2 identical sister chromatids joined at the centromere. In the cytoplasm, the centrosomes divide and move apart towards opposite ends of the cell. The second phase of mitosis is prometaphase. During prometaphase, the nuclear envelope breaks into fragments. A kinetochore has formed at the centromere of each chromatid, 2 per chromosome. The chromosomes begin to move toward the center of the cell at the metaphase plate. Some of the spindle fibers which grew from each centrosome reach the chromosomes and attach to kinetochores. The third phase of mitosis is metaphase. In metaphase, the nuclear membrane breaks off and the mitotic spindle is fully formed. The 4 chromosomes have all arrived at the metaphase plate, which is an imaginary plane that is equidistant between the spindle's 2 poles. The fourth phase of mitosis is anaphase. Anaphase begins when the 2 centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a daughter chromosome. The 2 new daughter chromosomes begin moving toward opposite ends of the cell as their kinetochore microtubules are attached at the centromere region. The cell begins to elongate as the non-kinetochore microtubules lengthen. The microtubules of the attached chromosomes shorten as the kinetochores walk the chromosomes toward opposite poles. The fifth and final stage of mitosis is telophase and cytokinesis. During telophase, the nuclear envelope forms around the identical sets of chromosomes at the 2 poles of the cell. The chromosomes begin to uncoil within each nuclear envelope. Cytokinesis begins shortly after as the cytoplasm splits. The cell membrane pinches to separate the 2 sets of chromatids. This process is known as cleavage. The first sign of cleavage in an animal cell is the appearance of a cleavage furrow, a shallow groove around the cell in the cell surface near the old metaphase plate. The chromosomes begin to unwind and become thin strands as originally structured. The cell is then separated into 2 daughter cells with the same number of chromosomes as the original parent. Mitosis results in 2 daughter cells. Simply put, mitosis is a process of cell duplication in which one cell divides into 2 genetically identical daughter cells. In the various stages of mitosis, the cell's chromosomes are copied and then distributed equally between the 2 nuclei of the daughter cells.